Again, thank you, Fernando, and thanks to all of you for showing up today. As Fernando mentioned, my lab is trying to create new life. It comes with a lot of different names. The most common one is xenobots. We'll talk about xenobots in a moment. We're going to talk about xenobots this morning and then talk about a very different research project going on in my lab known as polycomputation, and I'll talk about what that is in a moment. What these two seemingly very different research projects have in common, I believe, is that they point towards a very different future for AI, a very different way to go about trying to create autonomous, safe, and useful machines. We want all three of those. That is quite orthogonal to what's currently fashionable in AI, as we all know, which is token prediction. What other ways might we get to machines that are useful, autonomous, and safe? I think the xenobots xenobots and polycomputation, as you'll see in a moment, are examples of this. So what do I mean by a different approach to AI? If we pull way back and look at living systems, which of course is our sort of guiding star in trying to create intelligent machines, for the simple reason that we believe we are intelligent machines. So if Mother Nature created us, maybe we should take some hints from Mother Nature about how she created intelligent machines and try and build that into our machines. Biology has been an inspiration for AI and robotics since the very beginning in the 1950s. So we can make a lot of observations about living systems. The one I want to focus on today is that every single organism on this planet and every organism that has ever existed on this planet has a rich inner life, which is shown by my little cartoon here. What do I mean by a rich inner life? What I mean is that if you look at, for example, a human being at the whole body scale, you see sensors and actuators. You see eyes and ears and noses, the sensors, and you see the actuators. You see muscles and biceps and triceps. We, as an entire organism, are able to sense, think, and act. If you zoom in on a human being to the organ level, doesn't matter what organ you pick, the liver, the pancreas, that organ also has sensory apparatus. Your liver senses what's going on. Your liver, in a sense, thinks, and your liver does something. It acts. So when you zoom in on a human being to the organ level, you also find things that sense, think, and act. You can probably guess where the story is going. You zoom in again to the cellular level, and the cell is arguably the most complex machine on the planet. The surface of a cell is covered with sensory receptors. And cells are capable of very complex movements. They communicate with their neighbors. So an individual cell senses, thinks, and acts. And you can keep going. Subcellular assemblies, complex machines inside the cell, also sense, think, and act. So it's agents that sense, think, and act all the way down. That's what I mean in the rest of this talk when I talk about a rich inner life. In contrast, if you look at every single autonomous robot on the planet, autonomous vehicles, drones, they are sort of smart at the whole vehicle level. So an autonomous car, at the level of the car, it's got a camera on the roof. It's got four wheels with which it can do actions. It senses and thinks and acts. But if you zoom in on an autonomous vehicle, you immediately find things that don't sense, think, and act. You find inert materials, metal, plastic, and so on. That makes all of our robots, our current robots that do not have a rich inner life, they have a poor inner life, very dangerous. Because if the sensors at the whole vehicle level sense a situation that's far outside the vehicle's experience, there's no way for the vehicle to recover. There's no backup plan. A human being, you may be surprised. If a student who comes from near the equator and has never been on ice before and comes to visit us here at the University of Vermont, especially today, they may start to slip on the ice. At the whole organism level, that student is surprised. But the chance that every single part of their body is also simultaneously surprised, that probability is zero. There is some collection of cells, some small part of their body for which the local experience of slipping on the ice is not surprising, and that small bit of this poor student's body knows what to do, is not surprised, and because it's not surprised, it knows how to pull its neighbors who are freaking out back into their basin of familiarity. So what often happens is a young person who's never experienced ice before and they start to slip on the ice, they recover within a tenth of a second before they consciously even realize that they're recovering from slipping on the ice. And if you ask anyone who's been in that experience, it's miraculous. How did they know to recover their footing in a situation they've never been in before? The reason why is because human beings and every organism has a rich inner life. Something inside your body is not surprised by a surprising event and is able to pull the surprised parts back into non-surprise. That is the advantage of all organisms and something that no machines have. And the main driver in my lab at the moment of all of our research projects is how to create machines that do have rich inner lives, so that when there's external adversity, the real world is always surprising, it's always throwing new things at us. When a machine is faced with external adversity, it has previous experience with internal adversity. The bits inside the machine are constantly negotiating. They're constantly generating and taming internal adversity. And what my lab is in the process of showing is dealing with generating and taming internal adversity pre-trains machines, or can pre-train machines to deal with external adversity. Okay, that's the main message I wanted to send to communicate today. And I'm going to show you two examples now of technologies from my lab that have this ability to tame internal adversity and use it to handle external adversity. All right, so there's our guiding theory for today. Internal complexity, or the ability to deal with internal adversity, pre-trains life for handling external complexity or adversity or surprise. And we want to build that into machines. Okay, here we go. So let's start with the xenobots. What is a xenobot? Well, the name xenobot, we named these creatures that back in 2019 when my lab teamed up with Mike Levin's lab at Tufts. That's a biology lab at Tufts. At that time, the Levin lab was able to take about 2,000 cells from very early frog embryo, basically a frog egg. And when Doug Blackiston, a microsurgeon in the lab at Tufts, takes these 2,000 cells from frog egg, those 2,000 cells kind of ball up. They don't like being alone. They want as many neighbors as possible, so they kind of congeal up into a ball like you see here. So it turns out that this ball of about 2,000 frog cells, under certain conditions, will start to move, and we'll talk about the movement capabilities of xenobots in a moment. The first thing I wanted to show you about xenobots, first of all, I wanted to tell you what they are, is you can see immediately that this xenobot has a rich inner life. You can see the individual cells in this video. We shot this video by looking down through a microscope. The thing you're looking at is about 1 millimeter in diameter, about the size of a poppy seed. You can just see it with the unaided eye. These white flashes that you see, these are sudden increases in calcium concentration inside the cell. Cells communicate along lots of different channels. One of the primary channels they use for communication is calcium. It looks like there's kind of some random flashing going on here. And you'll notice that partway through this video, there's a massive flash of what looks like lightning. It's coming up in a moment. That is a response, the cell's collective response to Doug Blackiston, the microsurgeon, puncturing the xenobot with a very small pin. There's the puncture, there's the flash. You can see where the puncture occurred. This is something that these cells have never experienced before. These 2,000 cells are used to being part of a frog embryo. They are not used to being on their own. They're in a very surprising situation. And yet, these 2,000 cells don't die. They stay alive, they take in food, they excrete waste products, and when they're injured, you can see some of them migrating into the wound and they heal themselves up. This is an example of living internal complexity or rich inner lives, providing an adaptive response, the ability to maintain a situation, a shape, and a form very far from wild type, from what they're normally used to. Okay. This is what's known as a basal xenobot. And what we mean by basal xenobot is sort of the basic material. We take 2,000 cells from a frog egg. It's genetically unmodified cells. This is our living material. I mentioned, I just wanted to say, just to give you a sense of size, there's a xenobot on the right that's zoomed in, and you should be able to see this small black dot in the microscope slide on the left. That's what a xenobot looks like to the unaided eye. Okay, so why do we call them xenobots? Seems like kind of an odd name to give to a small piece of frog egg. Here's why. Back in 2019, when we teamed up with Levin and Blackiston at Tufts, they told us that they could take these cells from frog egg and rearrange them into new patterns. At that time, Doug Blackiston, our microsurgeon, could take 2 cell types from frog skin, which you're going to see represented in a computer in a moment as blue cubes. And Doug could also extract frog heart cells from a frog. These are like your heart cells. They spontaneously get bigger and smaller. And if you arrange frog heart cells into the shape of a frog heart or human heart cells into the shape of a human heart, those heart cells all hold on to one another and they all get bigger together and they all get smaller together, which causes your heart to act like a pump, which is very good news for you. So the heart cells, as you're going to see in a moment, are going to be used to become the motors of our robot. So at this point in 2019, we knew that Doug could take 2 types of Lego bricks, frog skin, which are soft and passive, like your skin, and frog heart cells, which spontaneously get bigger and smaller. And we asked the following question. If Doug can take these 2 types of cells from early frog and put them together in new shapes and sizes and new distributions, could we replace Doug with an AI? Could we ask an AI to find a way to put skin and heart cells together to produce a robot, something that moves around and does something useful on our behalf? Okay, so here we go. Here's our AI at work. So what we did in this very first experiment is we asked the AI to find a way to put skin and heart cells together to produce something that would walk along the bottom of a petri dish. The AI got to work by generating a whole bunch of random designs. This is a generative AI. What does this generative AI generate? It generates random combinations of skin and heart cells, as you can see in a simulator. So we built a simulator that is called a biophysical simulator. It approximates how these 2 cell types behave. You can see it's an approximation already because it's approximating each cell as a cube. This is like the Minecraft of synthetic biology. Okay, so I'm showing you 3 of the initial random designs. In this very first experiment, the AI actually generated 100 random designs. You're just not seeing the other 97. Not surprisingly, if you put skin, which are the light blue cubes, and red-green cubes, which are the heart muscle cells, together in random combinations, none of these 3 designs do what we ask the AI to get them to do, which is to walk along the bottom of the petri dish, shown in white here. They don't go anywhere. Turns out, though, that if you look very, very carefully, the video in the bottom, the xenobot in the bottom, is moving about 1 millimeter to the right. We actually asked the AI to make the bots move to the right along the bottom of the petri dish. So one of these designs is slightly less crappy than the other 2. So you can probably guess what the AI does. It deletes the 2 crappy designs, the top one and the middle one, and the AI makes a randomly modified copy of the survivor, the one simulated xenobot in the bottom. And those randomly modified copies, those offspring of that surviving design, look and move a little bit similar, look and move similarly to their parent, the design in the bottom right, but not quite the same. A couple of those offspring move to the right 2 millimeters rather than 1 millimeter. So in that next generation, the AI again deletes the slower moving xenobots and makes randomly modified copies of the faster moving bots. This particular AI is known as an evolutionary algorithm or a genetic algorithm for obvious reasons. That AI algorithm is based on biological evolution. Generate a population of diverse things, in our case, simulated xenobots, delete the ones that do a poor job, and allow the ones that survive to produce offspring. It's a very, very simple type of AI. It's been around since the 1960s. So far, so good? Okay, I'm providing a lot of explication. I don't know if anyone's asking any questions in chat. I'm happy to clarify anything. Yep. Yep. Yeah. So, great question. So what Doug actually did, I'm going to get to the edge of my biological expertise here. He harvested motor stem cells. So stem cells that were destined to become muscle or heart, and they end up becoming heart. And then what he harvested from the embryo were skin stem cells. So they're stem cells. They're not stem cells. They're not totipotent. They're partially potent. They are going to differentiate into the different kinds of skin. And it was actually unknown. Yep, go ahead. A little bit, a tiny bit, but mostly it's done at that point. Most of the division has ended. I think there's a little bit, but it's negligible. Okay, good questions. Okay, so we got some biologists on the call. Good to know. Okay, so so at this point, this is another question. Okay, no problem. No problem. So, any questions? Yep. Everybody hear me okay? Okay. Okay, so the process I'm describing right now, this is an AI algorithm running simulations on the supercomputer here in Vermont. So the biology is paused at this point. We ran this evolutionary algorithm for about 2 weeks on the supercomputer, and at the end of those 2 weeks, the AI gave us back this design. Basically, the AI just emailed us this MP4, this video. So here you can see that the AI has evolved a design. It's evolved so that there is there's heart tissue on the bottom half of the bot and skin on the top. I'll mention something about the red-green flashing. Red is when the simulated heart cell is getting bigger, and green is when the simulated heart cell is getting smaller. You'll notice that the red-green flashes are not synchronized. Again, luckily for you, heart cells tend to synchronize when in the shape of a heart. We asked Doug Blackiston, our microsurgeon, what would happen if you put together heart cells like this, and he said, I have no idea whether they'll synchronize or not. So in the simulator, we didn't allow the simulated heart cells to synchronize, which you can see here. So one of the things that was exciting to us when we got back this design is we got a machine that's pretty reliable. It moves more or less in a straight line to the right, which is what we wanted. It reliably moves right, but the parts that it's made up from are unreliable. They don't synchronize. This is a notoriously difficult thing for human engineers to do, is to build a reliable machine out of unreliable parts. It's not impossible, but it's very, very difficult. So again, another cool thing for us is that here's a demonstration of AI doing well at something that is really, really, really hard for humans. Okay, that was the end of part 1 of our first Xenobot experiment. Part two was back to the biology. So now we're again looking down through the microscope with Doug, our microsurgeon. At this point in the construction process, he's liberated some heart stem cells from frog embryo, and you can see the individual cells here. He's deposited these cells in a little small indentation in the bottom of the petri dish. So they're kind of settling into that indentation in kind of a carpet, almost a 2D sheet of cells. So this is Doug trying to follow the AI generated blueprint. Remember, heart on the bottom. So Doug at this point has managed to create a little layer of heart. Later, he sprinkles another layer, another bunch of heart cells, and they kind of settle in another layer on top, another layer of heart. Then Doug sprinkles some skin cells, and they settle into a layer of skin on top. And Doug is basically kind of building up a sandwich with layers according to what the AI wanted. Heart on the bottom, skin on the top. Once he's put down these layers, these cells are very adhesive, and they stick to one another. This is a lower magnification, so we're kind of zooming out. You can no longer see the individual cells. There's about 2,000 cells here, and you can see that Doug is using very, very small, basically tweezers, to scrape away the unwanted material and to make these little stubby legs that the AI discovered. So again, basically this is just sculpture, something humans that have been doing for thousands and thousands and thousands of years. Very old technology, or a craft, I guess, in this case. So this is Doug in vitro with real cells trying to approximate the shape and the cellular distribution that the AI came up with. And that's the end of stage two. So at the end of stage two, what Doug is going to now do is take this little xenobot, put it right side up, so its little stubby legs are on the bottom of the petri dish. And in phase three here, we crossed our fingers, hoping that the in vitro bot, which you see in the bottom, approximated the behavior of the in silico bot, the simulated bot that you see in the top. And you can see that, sure enough, both of them more or less move to the right. The in vitro bot doesn't quite have exactly the same shape that the AI wanted it to, and it doesn't quite move in the right way, but more or less it does what it more or less moves in the way that the AI predicted that it would. That's the end of our first experiment with xenobots, and part of the reason why this all worked is because frogs have rich inner lives. No frog has ever been in this shape before, in about this millimeter-sized shape with back stubby legs. This is very far from the experience of frog eggs, frog embryos, frog tadpoles, and adult frogs. But despite being very far from its comfort zone, this collection of 2,000 cells holds together. They don't die, and they continue to, or the heart cells at least, continue to get bigger and smaller, and they reliably exhibit the behavior that we wanted. So among other things, this demonstrates that we can use AI to push living materials very far from wild type. In other words, we can get AI to build whole new kinds of organisms that look and act very different from any organism that's existed on Earth, at least as far as we know, and definitely looks and acts very differently from the host organism, which in this case is frog. Okay, I'll just pause there for a moment. I'm not sure if there's any other questions. Kimball has a question. Yeah, thank you, Kimball. It's a really good question. So what you're describing is actually a whole new field called biohybrid robotics. And it's exactly what you said, which is in some cases you want to build a machine where some of the parts are built from technological components, metal, plastics, batteries, circuits, and other parts of the same machine are built from biological components like cells and organs, for the simple reason that inorganic materials have their pros and cons, and organic materials have different pros and cons. And wouldn't it be great to build a machine in which you can bring together the best of both worlds? It's it's a brand new field. They just had their first conference this past summer in Switzerland, and I managed to attend. So great that you're thinking about that. It's a whole new way of thinking about robotics. It brings me to another point I wanted to point out, which is a lot of folks, when they learn about the xenobots for the first time, they say, this is ridiculous. That's not a robot. It's a frog, or at least it's something like a frog. It's important to remember that the original definition of robots comes from a Czech playwright who wrote a play called Rossum's Universal Robots over 100 years ago now. And in that play, these artificial men that were doing work on behalf of their human masters, they were made from some weird biological protoplasm. So it was only sort of in the 20th century, when we got really good at working with steel and eventually electronics and all the rest, that the general public has come to equate materials with robots. Robots are defined not from what they're made from, but what they do, which is typically move around on their own, which xenobots do. And robots are designed by us, in this case, designed by an AI, where the AI was designed by us. They're machines that are designed by us to do something useful on behalf of humans. That's the definition of a robot, and that's why I'm comfortable at least referring to these things as xenobots. The xeno part comes from the Latin name of the specific frog that donated cells to this project, which is Xenopus laevis. And xeno also means newcomer or stranger in Greek. So we thought that was also kind of cool. So that's where xenobots, the name, came from. How would AI be integrated into living tissue? Is that the question? Yep. Okay. That is a really good question. So obviously here the AI is outside. It's running on electronics in our supercomputer. It's definitely not inside. It's definitely not inside. It's not inside the xenobot. So it's a good question. How would you put AI inside a xenobot? Okay. All right. So now we're straying into philosophical material. Are they intelligent? Let me answer that question by jumping ahead a few slides to try and answer this. Or at least let me give you food for thought, and you can try and answer this question yourself. So I already showed you this video. This is, again, a xenobot made up of about 2,000 genetically unmodified cells. This thing, under certain conditions, will move around. As you can see here, it recovers from an injury. You know, is it intelligent? Is it thinking? That's kind of a philosophical question. However, one of my postdocs, Thomas Varley, conducted an interesting experiment. He took that xenobot that you just saw and some other ones and very carefully measured the amount of calcium in each of the 2,000 cells, which produces time series data like you see here. So we have time on the horizontal axis. On the vertical axis, we have one line for each of these 2,000 cells. And up, when the line goes up, that's calcium inside the cell becoming more concentrated. And when the line goes down, that's calcium becoming more dilute in the cells. Okay, nothing controversial here. This is just measurement. It turns out that if you then take each of these lines, you can ask how correlated they are. So two lines that are correlated, they either go up together and go down together, or when one line goes up, the other line always goes down, and when that line always goes down, the other line always goes up. Those are two correlated time series signals. If they're uncorrelated, there's obviously no pattern between them. So what you see on the right is a 2,000 by 2,000 figure, and every pixel, the pixel at position ij, is reporting the amount of correlation or covariance between two cells. A red pixel means they're anti-correlated. Whenever signal I goes up, signal j goes down. A blue pixel says that they're positively correlated. Did I get that right? Red, blue, doesn't really matter. Oh, sorry, it's the other way around. But anyways, where you see a white pixel, that means there's no relationship between those two signals. Turns out if you then shuffle these signals, you get these 1, 2, 3, 4 red blocks. So there are pieces, there are subsets of cells within the xenobot that are co-varying. It's impossible to see by eye. when I showed you that video, for most of you, presumably you just saw random, non-correlated behavior. It turns out that there's underlying structure here. It turns out that that underlying structure is almost indistinguishable from pattern time series data inside the human brain. If you take someone and put them inside an fMRI machine and you measure 2,000 different places inside their brains, you see more or less the same kind of patterns. So if you watch electrical flashing inside a brain or calcium flashing inside a xenobot, they look very, very, very similar. Most of us are comfortable with saying human brains think. So if you're okay with human brains thinking, and human brains tend to exhibit the same sort of patterns as xenobots, it's kind of hard to not also conclude xenobots are thinking. I am not arguing that you should think so, but it seems quite compelling. So there's a lot of things about xenobots that I think are pretty cool. One of them is they kind of, they're demolishing some of the things that we were taught in high school. It turns out we were lied to in high school. In high school, you were probably taught that frog DNA encodes for frog and human DNA codes for humans. Turns out that frog DNA also encodes for xenobots. So DNA is not a blueprint for frog phenotype, for example. The other thing you were probably taught in high school, or at least the general culture, teaches us that brains are the seat of intelligence. That's where thinking comes from. This particular work by my postdoc and some of our other colleagues suggests that maybe brains are not the only seats of intelligence or thinking. It's possible that this clump of 2,000 frog skin cells is also thinking. I don't know what to make of that fact, but there you go. That's a really good question, Madison. And I think actually it's more the latter. So we have some research now where we're doing exactly that, which is the AI generates electrical fields. And those electrical fields try and quote unquote convince the cells to form up and act in the way we want them to. So at the moment, xenobots are made in this top-down design manner. Doug comes in with his knives and his forceps and creates what he wants by force, by mechanical force, which is great, but it's kind of a blunt weapon, not the best way to do things. So can we convince cells externally with things like electromagnetic fields? If it is possible, it's a very complicated thing to do, and therefore it's probably something that an AI can probably learn to do better than we can. And that's something we're working on at the moment. Great question. Yeah, that's a really great question. We don't have one goal. At this point, I think we have dozens of goals, and the list is getting longer all the time. But there are there's a couple of main sort of buckets of goals. The first one, which I'm most interested in, is the basic science one. So I just mentioned that, this idea that DNA encodes adult phenotype and brain is the seat of intelligence. I'm interested in science that kind of overturns our common assumptions. So I'm interested in pursuing future experiments with xenobots that help us really understand, what is DNA doing if it's not encoding adult phenotype? If there are other seats of intelligence other than brains, where and how? Is the xenobot actually thinking or not, and what does that mean? And there's many, many questions related to that. So there's a lot of basic science that I hope we get to work out in the years to come, given this technology. Then there are engineering goals. So for example, you might have a Roomba at home that just goes around and, cleans things up. We're trying to basically make xenobot Roombas. So xenobots are frog cells, so they're happiest in room temperature fresh water. So if Roombas can clean dirt off your floor, could we make xenobots that clean microplastics out of, your waterways, your tap water? And then further, could we create machines? Could we engineer machines that not only take microplastics out of the water, but take cancer cells out of your bloodstream. So I don't have time to talk about it here, but we have a growing set of papers around anthrobots. These are biological robots that are made from human cells. So I think there will be medical applications. They're quite a number of years off in the future, but it could be possible that we could create machines that operate inside a patient's body, and those machines are made from the patient's own cells, which would sidestep immune response problems. So sort of engineering / medical applications as well. The last sort of goal I wanted to mention about, this is kind of going back to the scientific goals, is what kind of life is out there? So I don't know if we'll all live long enough to discover aliens, but it turns out that AI can create new kind of life forms that are very different from those that currently exist on Earth. How far can AI go from the wild type donor species? So if you think about this concept of morphospace, the space of all possible organisms, current organisms that exist on Earth, current species, occupy a vanishingly small set of islands in that overall morphospace. So we could ask an AI, we could say, put frog behind you in morphospace and design creatures in the other direction. Go as far away from frog as you can. Try and make something that is most unlike frog as you can, or most unlike other organisms, and what is possible, what can be created. So we may not find aliens soon, but maybe we can create them. I think that would be a goal worth pursuing. Those are both good questions. Processing memory, we have absolutely no idea, like, at least for that xenobot in the upper left, there are 4 parts within that actual xenobot that are correlated. Those are the red squares there. What is it that those cells are doing? Are they talking to one another? Are they planning? Are they trying to incorporate a memory that they're going to act on later? Is it just like some crazy coincidence? Like, are they not communicating? They're not processing. They're not remembering. They're not planning. It's just, I don't know, some sort of weird statistical thing. This is, again, going to be one of the fun things over the next few years, is to try and actually figure out what if any, these cells are doing when they become correlated like this. One other wrinkle I'll mention is that not only are there these 4 sets, but they often also become decorrelated for a while. They sort of fall apart as a component and then resynchronize again. So it's not that it's not a xenobot that's built from 4 big parts and those parts exist throughout the lifetime of the xenobot. They kind of seem to come into existence and then fade back out again. What they're doing, if anything, I have absolutely no idea yet. That's a great question. So could we make a xenobot that attaches to a very small 3D printer to make more? The work that I talked about today, this was our first published paper on xenobots back in 2020. If you Google my last name and 2021, you'll find our 3rd paper, which was published in 2021. And in that paper, we showed that xenobots kinematically self-replicate. Not using a small 3D printer, what those xenobots do, I'll try and say this in 30 seconds. If you put a bunch of xenobots in a petri dish and you sprinkle additional individual frog cells, those bots will act like Roombas and they'll just kind of push the cells into piles. And those piles of pushed together cells, because they're adhesive, they adhere and they become an additional multicellular assembly in the dish. It's another clump of cells. And some of those clumps grow very small hairs on their surface called cilia. You have cilia in your nose, in your bronchial cavity to get rid of dust and all that stuff. And it turns out that in frogs, those cilia beat against the fresh water, and some of these piles grow enough cilia that the pile itself starts to move around and pushes other cells into piles, which produces yet more xenobots and yet more xenobots. That's why we call it kinematic self-replication. The bots make copies of themselves through kinematics, through movement. They're not accurate copies of themselves. I think you mentioned, like, can you make more accurate ones? So you lose replicative fidelity. Like, they kind of the piles become smaller and messier and move slower, and eventually the process runs out of gas. But you can, again, the fun is like finding things that life can do that you tend not to see in nature. You don't see kinematic self-replication in living machines. Xenobots seem to be able to make copies of themselves in this way. Thanks so much, Fernando. Thanks, everyone. Bye bye.